Assalamualaikum. My name is Sharifah Anil Akilah binti Syed Adnan and my partner is Nabihah binti Helmi. Today we're going to present case study about textile wastewater characterization and its reduction of its COD and BOD by oxidation. First, we go to practical 23, determination of chemical oxygen demand in wastewater. In this experiment, COD method was used to measure the pollution strength of wastewater. Potassium dichromate is chosen due to its strong chemical oxidizing capability. Closed reflux method was used in this experiment due to the small quantities of samples. Through this experiment, the protocol in conducting chemical oxygen demand tests and the correlation between chemical oxygen demand and biological oxygen demand in wastewater was determined. Next, we move to our case study, textile wastewater characterization and reduction of its COD and BOD by oxidation. In Pakistan, textile industrial is one of the most largest and important industrial sectors. However, textile industry can be affected by environmental problems, mainly caused by discharges of water. In this case study, the main goal was to characterize textile wastewater to evaluate the treatment options, evaluate the efficiency of 5 oxidizing agents in the treatment of wastewater, and optimization of the best operating conditions for oxidation process.
Hello and Assalamualaikum Prof K. My name is Nabihah binti Helmi, metric number A174235. So my part is about scientific review. For the first video, the title is Chemical Oxygen Demand, COD. The video differentiates about chemical oxygen demand, COD, and biodegradable oxygen demand, BOD, in aspects of time requirement for these two methods to take place, ability to oxidize organic matter. They also discuss about calibration curve to determine the relation between COD value and absorbance. Besides, they discuss further more in preparation of sample, handling the dry box digester, measure the sample using spectrometer, and calculate the COD referring to calibration curve. The second video is about COD measurement. The video shows us step-by-step handling the experiment. First, how to prepare the blank samples and samples. Then, how to run the digestion method. This table will be used to compare two experiments from the video and from the case study. Experiment from the video was determined by using closed reflux method because only involving a small amount of sample. Meanwhile, from the case study, we are using open reflux because textile production involves a number of wet processes that use a large amount of water sample. Meanwhile, for determination method, the experiment from the video used spectrophotometric to determine the COD measurement. Meanwhile, for the case study, we are using tachymetric method to measure the COD and BOD. There are 5 oxidizing agents contributed in the case study, which is potassium dichromate, hydrogen peroxide, sodium hypochlorite, calcium hypochlorite, and also calcium dichromate. Meanwhile, for the video, we are using only one oxidizing agent, which is potassium dichromate. The differences at the process between these two experiments occur at digestion method. There are 3 variables in the case study, which is retention time, temperature, and oxidizing agent. So basically, during digestion method, samples were heated with different temperature, 25 degrees Celsius, 50 degrees Celsius, and 100 degrees Celsius, and retention time 5 minutes, 90 minutes, 1440 minutes recorded for each temperature. Amount of oxidizing agent required for oxidation were calculated by using stoichiometric calculation for the case study. Meanwhile, the procedure for the video, first we are prepare the sample and blank sample, same as the case study, and then reduction by oxidizing agent and adding the sulfuric acid, and undergo digestion for 2 hours at 150 degrees Celsius, and observe the color, and then measure the COD measurement by using calibration curve. This table represents the complete picture obtained from the textile wastewater sample analysis. The most frightening values are for COD, BOD, and heavy metals such as chromium, cuprum, and zinc that cause a real threat to the environment. This is the graph about the effect of retention time on COD removal at 25 degrees Celsius. So, as we can analyze the whole graph, the maximum COD reduction was achieved by calcium hypochlorite at 25 degrees Celsius compared to the other 4 oxidizing agents. COD reduced from 400 mg per liter to 154 mg per liter. And the minimum reduction of COD was achieved by hydrogen peroxide from 400 mg per liter to 273 mg per liter at retention time 1440 minutes. So overall for the graph, COD reduction rate decreased over the period applied for all experimented oxidizing agents. The next graph is about effect of retention time on BOD removal at 25 degrees Celsius. From the graph, the maximum BOD reduction was achieved by calcium hypochlorite reduced from 300 mg per liter to 147 mg per liter, 125 mg per liter, and 78 mg per liter at retention time 5 minutes, 90 minutes, and 1440 minutes respectively. BOD reduction rate decreased as retention time was decreased 5 minutes, 90 minutes, and 1,440 minutes for all oxidizing agents, as you can see in the graph. For the third graph, it is about the effect of retention time on COD removal at 50 degrees Celsius. So, when the temperature reached at 50 degrees Celsius, it was observed that calcium hypochlorite is still the maximum oxidizing agent that reduced COD from 400 mg per liter to 200 mg per liter at retention time 5 minutes and continue to reduce to 158 mg per liter at retention time 1440 minutes. Then, the minimum reduction of COD by hydrogen peroxide before the treatment 400 mg per liter and to 270 mg per liter and 250 mg per liter at retention time 5 minutes and 1440 minutes respectively.
Next, we move to figure 2b, effect of retention time on BOD removal at 50 degrees Celsius. In this graph, it shows the value of BOD reduction is decreasing as the time increasing. The value of BOD reduction at 50 degrees Celsius is lower compared to the value of COD reduction at 50 degrees Celsius. The value of BOD reduction achieved by calcium hypochlorite is the lowest. Next is graph of effect retention time on COD removal at 100 degrees Celsius. Again, the value of COD reduction is decreasing as time increasing. The minimum value of COD reduction also was achieved by calcium hypochlorite. For the final graph, compared to the RDS graph, the value of BOD reduction at 100 degrees Celsius is the lowest, which is below 100 mg per liter. The value of BOD reduction also inversely proportional with time. Through this case study, we can conclude that the maximum value of COD and BOD reduction was achieved by calcium hypochlorite at 100 degrees Celsius. Both COD and BOD test methods are used to give an indication of the amount of pollution in a water sample. COD is the amount of oxygen required to chemically break down the pollutants, whereas BOD is the amount of oxygen required to do this biologically through microorganisms. The value of COD is always more than BOD because COD method measures the total oxygen, including inorganic and organic oxygen content, while BOD represents only the amount of oxygen required to degrade the organic matters present in wastewater by the microorganisms.
So for the question, why the COD analysis and BOD analysis give different results for the same waste? Because the COD tests oxidize both biologically degradable and unbiodegradable organic material, meanwhile BOD only oxidize unbiodegradable organic material. So the energy available for biological action is always overestimated. That means COD value always greater than BOD value. However, we does not reduce the usefulness of the test. If it is assumed that the fraction of organic material that is not oxidized in the COD test remain constant, then any change in COD between 2 points in the processes provides an assessment in terms of oxygen of corresponding energy change. The change in COD then can be used to establish the kinetic of energy conversion in the process. For example, the energy removal can be directly linked to the COD change. By contrast, BOD value requires a correction factor to correspond to energy change because the test value do not reflect the total oxygen demand.